This is Chapter 17, Module 2, Ears and Hearing. The learning objectives of this module are 1. Describe the structures of the ear and their roles in the process of hearing, and 2. Describe the structures and processes involved in maintaining equilibrium. In the external anatomy of the ear, the auricle or pinna is the outer fleshy and cartilaginous part of the ear. This area protects the opening of the auditory canal, and it also directs sound waves into the auditory canal. The external acoustic meatus, or auditory canal, is a passageway to the middle ear. At the end of the external acoustic meatus is the tympanic membrane, or eardrum. The tympanic membrane is a thin, transparent sheet that separates the external ear from the middle ear. It is very delicate. Ceruminous glands are along the auditory canal, and they secrete a waxy material called cerumen or earwax. Cerumen helps to slow the growth of microorganisms. The middle ear is an air-filled space deep to the tympanic membrane. Within the middle ear is the auditory tube, which is a passageway leading from the middle ear to the nasopharynx and the mastoid air cells. This auditory tube equalizes pressure on either side of the eardrum. An infection in the middle ear is called otitis media. In the middle ear are 3 tiny ear bones called auditory ossicles. These ear bones connect the tympanic membrane to the internal ear. The names of the bones are the malleus, the incus, and the stapes. Sound waves will cause the eardrum to vibrate, which in turn causes the ossicles to vibrate, which in turn causes the inner ear to vibrate. There are 2 small muscles that protect the tympanic membrane and the ossicles from loud noises. The Tensor Tympani muscle originates on the temporal bone and inserts into the malleus. This muscle will stiffen the tympanic membrane. The Stapedius muscle inserts into the stapes bone. This muscle will reduce the movement of the stapes at the oval window of the inner ear. The internal ear is deep to the middle ear. The bony structure in the inner ear is the bony labyrinth. The walls of the bony labyrinth are continuous with the temporal bone. Inside the bony labyrinth is a membranous labyrinth containing a membranous tube. The space in between the bony labyrinth and the membranous labyrinth is filled with a fluid called perilymph. The space inside the membranous labyrinth is called the endolymph. The bony labyrinth can be divided into the vestibule, the 3 semicircular canals, and the cochlea. The vestibule consists of a pair of membranous sacs. The saccule and the utricle, which provide sensations of gravity and linear acceleration, are found within the vestibule. The semicircular canals enclose the membranous tubes called the semicircular ducts. Receptors in the semicircular ducts are stimulated by rotation of the head. The vestibule and the semicircular canals together are called the vestibular complex. The cochlea is a spiral-shaped bony chamber that contains the cochlear duct of the membranous labyrinth. Receptors within the cochlear duct provide the sense of hearing. The bony labyrinth surrounding the cochlea consists of dense bone except at 2 areas that are membranous. The round window separates the perilymph of the cochlea from the middle ear. The oval window is found under the stapes. Equilibrium is a sensation that is provided by the receptors of the vestibular complex. The semicircular canals provide information about rotation of the head, nodding yes or no, or laterally bending your head. The receptors in the canals give information about how rapid the movement is and in what direction the movement is. The saccule and utricle convey information about your position with respect to gravity. If you stand with your head tilted to one side, these receptors report the angle involved and whether your head tilts forward or backward. The saccule and utricle are also stimulated by acceleration and deceleration. When your car accelerates from a stop, the receptors in the saccule and utricle give you the sensation of increasing speed. The semicircular canal contains 3 separate ducts, the anterior, posterior, and lateral semicircular ducts. Each semicircular duct contains an ampulla, which is an expanded region that contains the receptors. The wall of the canal where the receptors are found is called the crista. Within the crista are receptors called hair cells. An external force can bend the hair cells, which release chemical transmitters when stimulated. Each crista is bound to a cupula, which is a gelatinous structure that extends the full width of the ampulla. When your head rotates, the endolymph fluid in the semicircular ducts moves along the semicircular duct. When you shake your head no, the hair cells of the lateral semicircular duct are activated. When you nod your head yes, the hair cells of the anterior semicircular duct are activated. And when you tilt your head from side to side, the hair cells of the posterior semicircular duct are activated. The utricle and saccule provide information on whether the body is moving or not, as well as equilibrium. The hair cells of the utricle and saccule are clustered in oval structures called maculae. The hair cell processes are embedded in a gelatinous material. The surface of this gelatinous material contains dense crystals called statoconia. When your head is in the normal upright position, the statoconia sit atop the macula. The weight of the crystals pushes the hair cells down rather than to the side. When you tilt your head, the pull of gravity on the statoconia shifts them to the side, therefore distorting the hair cell processes. This stimulates the hair cell to tell the central nervous system that your head is no longer level. The sensory neurons that are activated by the hair cells will send action potentials to the brain. The sensory information will reach the brainstem and then cross to the cerebellum. The receptors for hearing are found in the cochlea. The cochlea is a spiral structure that is filled with a perilymph fluid. The cochlea has a membranous duct inside its bony labyrinth. This duct is called the Cochlear Duct. The Cochlear Duct divides the bony labyrinth into two ducts: the vestibular duct, known as the scala vestibule, and the tympanic duct, known as the scala tympani. The outer surfaces of these ducts are the bony labyrinth. The vestibular duct is continuous with the tympanic duct at the tip of the cochlear spiral. The vestibular duct starts at the oval window and extends to the tip of the cochlear spiral, and then continues back to the round window as the tympanic duct. The cochlear duct is the structure that divides the vestibular duct from the tympanic duct. The hair cells of the cochlear ducts are located in the organ of Corti, a structure also called the spiral organ. The organ of Corti sits on the basilar membrane, which is a membrane that separates the cochlear duct from the tympanic duct. The hair cell extensions are in contact with the tectorial membrane, which lays over the top of the hair cells. The tectorial membrane is firmly attached to the inner wall of the cochlear duct. When a portion of the basilar membrane bounces up and down, the hair cell extensions are pressed against the tectorial membrane and become distorted. The basilar membrane moves when fluctuations within the perilymph move it. The pressure fluctuations within the perilymph are triggered by sound waves that vibrate the tympanic membrane. Hearing is the perception of sound. The pitch of sound is our sensory response to its frequency of a sound wave. Amplitude of sound is our sensory response to the intensity of a sound wave. In the hearing process, the sound waves enter the external acoustic meatus and travel toward the tympanic membrane. The tympanic membrane vibrates from the sound waves and causes the displacement of the auditory ossicles. That is, when the tympanic membrane vibrates, so do the malleus, the incus, and the stapes. The stapes is connected to the oval window, and when the stapes vibrates, it causes the oval window to bulge. This action causes pressure waves in the perilymph fluid on the other side of the oval window, in the vestibular duct. The pressure waves that were created in the perilymph fluid in the vestibular duct will distort the basilar membrane as it comes in contact with the cochlear duct. The pressure waves first distort the vestibular membrane of the cochlear duct, and then the basilar membrane of the cochlear duct. The location of the distortion is different for every frequency. If the frequency of the sound is high-pitched, it has a very short wavelength and will distort the basilar membrane closer to the base of the cochlear duct. If the frequency of sound is low-pitched, it has a long wavelength and will distort the basilar membrane closer to the tip of the cochlear duct. When the basilar membrane becomes distorted, it causes a pressure wave in the tympanic duct. This pressure will travel in the tympanic duct toward the round window. The round window will bulge out when the pressure wave hits it and then helps to absorb that pressure. The vibration that occurs in the cochlear duct will cause the basilar membrane to vibrate. This vibration moves the hair cells against the tectorial membrane. The extensions of the hair cells bend as the hair cells move against the tectorial membrane. Stimulating the hair cells by bending them will result in the opening of sodium-gated channels in the hair cells. This causes depolarization of the hair cells, leading to an action potential in the axon and a release of neurotransmitters. The release of neurotransmitters will stimulate the sensory neuron. The sensory neurons are bipolar sensory neurons. A very soft sound may stimulate only a few hair cells. With more intensity, more hair cells will become active. The number of hair cells responding give information to the intensity of the sound. The action potentials in the sensory neuron will be relayed to the central nervous system over the cochlear branch of the cranial nerve 8, vestibular cochlear nerve. Cranial nerve 8 then synapses in the medulla oblongata onto a neuron that delivers the information to the thalamus. At the thalamus, another synapse occurs and the information is then delivered to the auditory cortex and other regions of the brain. This ends Chapter 17, Module 2, Ears and Hearing.